Look closely at the world around you, and the sheer volume of living things becomes overwhelming. To date, scientists have discovered and described roughly 1.7 to 1.8 million unique species on Earth. But dealing with that many organisms creates a massive communication problem. If you rely on local names, the same plant or animal will be called something completely different depending on the town, country, or language you're in. To eliminate this chaos, the scientific community needed a single, standardized way to identify, name, and categorize every living thing they found. Without an agreed-upon language of life, comparing a biological discovery made in Brazil with one made in Japan would be impossible. Taxonomy solves this through the scientific process of sorting organisms into convenient, recognizable categories based on their observable physical and cellular characteristics. This systematic approach relies on 4 sequential steps. First, we characterize the organism's traits. Second, we identify it. Third, we classify it into a group. And finally, we assign it a formal name, a process known as nomenclature. The globally accepted standard for this is binomial nomenclature. It provides every known organism with a unique 2-word scientific name. This approach to systematic arrangement traces its roots back centuries to the work of Carolus Linnaeus, who formalized the method in publications like Systema Naturae. The rules of this naming system are strict. The names are derived from Latin. The first word dictates the genus and is always capitalized. The second word is the specific epithet, or species, and is written entirely in lowercase. Providing a specific, globally recognized binomial name ensures precise communication. A biologist in Kenya and a botanist in Canada can look at the same Latin label and know exactly which organism they are discussing. Naming an organism is only half the job. Every identified species is also placed into a massive, tiered filing system. Each rank within this biological ladder is called a taxon. At the very top sits the kingdom. This is the broadest category, grouping together vastly different life forms that share only the most basic, fundamental characteristics. Below the kingdom, we filter organisms down through increasingly specific ranks: phylum, class, order, family, and genus. The core rule is simple. The lower you go on the ladder, the more physical and genetic traits members share. At the absolute bottom is the species level. This tier isolates specific organisms with fundamental similarities, distinguishing them clearly from their closest relatives. This hierarchy provides a precise, scalable address for every living thing, built entirely from its distinct traits. Let's see how this filing system applies directly to human beings. We start in kingdom Animalia. Having a spinal cord filters us into phylum Chordata. Body hair places us in class Mammalia. We move down into order Primata, then family Hominidae. Finally, at the bottom, we reach our binomial name, genus Homo, species Sapiens. Organizing humans within this exact framework shows that we do not sit apart from nature. We are subject to the same biological organization as a potato, or a housefly, or a galloping horse. Taxonomy provides the structural foundation for systematics, the study of evolutionary relationships between organisms. When you classify life both horizontally across related groups and vertically down the hierarchy, you realize that all present-day organisms are linked to creatures that lived on this earth millions of years ago. Evolutionary biologists like Ernst Mayr spent their careers pushing this realization forward, proving that the origin of species diversity is the central question of modern biology. Understanding the living world through this classification maps an unbroken family history, humbling humanity by proving our direct connection to millions of other species.